Hello, good evening, students. Today we are going to talk about the chromosomes. Okay, before going to talk about the chromosomes and all these things, I mean, how to study the chromosomes as well as the different types of chromosomes. For that, what we have to do is we have to need to prepare a karyotype. So, what actually you mean by a karyotype? Now, karyotype is nothing but it is a summary of the chromosome constitution of a cell or a person, such as 46XY chromosome. So, karyotype is nothing but it is a summary of the total chromosomal constitution of any cell. The cell it may be the cell of any person or any animal or any what do you say the organism. So such as for example in human beings we have 46 chromosomes. So we call it as 46XY. So 23 pairs of chromosomes are there. So karyotype actually it indicates what? It indicates nothing but a summary of the chromosomal constitution. Now it is actually used to mean an image showing the chromosomes of a cell which are sorted out in order and arranged in pairs. So in general, we can say that karyotype is nothing but a summary of the chromosome constitution of any cell, okay, of any human being, or it may be of a what is it, any organism, that is especially the animals. And here the chromosomes actually are arranged in the form of an image which shows the chromosomes arranged in an order and they are arranged in pairs. So such a what do you say the summary or an image you call it as nothing but a karyotype. Now apart from the karyotype, you have what? The karyosome. Karyogram. Karyogram is nothing but a display of the chromosome of a cell which is again sorted out into pairs. So we can differentiate now the karyotype and karyogram to be. Karyotype is only a summary, okay, which means an image. It is only an image which shows the chromosomes of a cell that are arranged in an order in pairs. Whereas karyogram is nothing but a display of the chromosomes. Okay. Now this is how the chromosomes that are arranged. Okay. Now here you can see the different types of chromosomes being arranged. Now the chromosome, if you see the structure of a chromosome actually has got two arms. One is called as a P arm and the other one is called as a Q arm. And in the center, you have a centromere or the kinetochore. So based on the position of the centromere, actually they are classified into different types. That will come to that later. One is metacentric. If it is in the center, present in the center, you call it as metacentric. Then submetacentric means it will be a little above. Okay. And then you have subacrocentric, means towards the what do you say, the tip. Okay. And acrocentric means almost near to the tip. Now telocentric means totally towards the tip. The centromere actually when it is towards the tip, then you say it is a telocentric. So like that you have different types of the chromosomes based on the position of the centromere. They have been classified. Okay. Now here they have given the example of the human chromosome groups. Okay, so they have been named into different types. You all know actually the humans consist of 23 pairs of chromosomes. Among these 23 pairs, 22 are the autosomes and 23rd chromosome is the allosome or the sex chromosome. So you have in human beings 22 pairs, total 23 pairs. Among the 23 pairs, 22 pairs are the autosomes and the 23rd pair is nothing but the allosome or the sex chromosome. Now these autosomes actually they are numbered from the largest to the smallest. So here they are seeing the chromosomes, like they have included chromosome group A includes 1 to 3 chromosomes, whereas they are the largest chromosomes and 1 and 3 are the submetacentric, but the 2 is 1 and 3 are metacentric and the 2 is submetacentric. So like that they have arranged. So this is actually done based on the karyotype study. So without karyotype and without actually preparing the chromosome plate, we cannot actually exactly find out the shape of the chromosome as well as the sizes of the chromosomes. So to study the chromosomes, not only of the humans, but any organism, if you take for example, okay, you need to prepare a chromosome plate. So here they have given, so B group consists of 4 and 5 chromosomes, C 6 to 12 chromosomes and 1 X chromosome, D consists of 13 to 15, like E consists of 16 to 18, F 19 to 20, and G 21, 22, and the Y chromosome. So like this is actually about the human chromosome group. So they have been classified or categorized into different groups from A to G. Now, next coming to the euchromatin and heterochromatin. So what do you mean by euchromatin and heterochromatin? So see, when you are actually going for the staining of the chromatin, now the chromatin actually shows two different types or two different regions. So based on the staining, they have been classified because one will be stained less and the other will take the stain more. So based on the staining mechanism, they have been classified into euchromatin and heterochromatin. Now, what does euchromatin indicate? Euchromatin is nothing but a part of chromatin which takes less stain. So when you are actually going for the staining mechanism, staining what do you say, the chromatin, euchromatin will take up less stain, okay, and it will be loosely packed and genetically active, involved in active transcription, and it is dispersed, showing dispersed appearance with more amount of DNA content than RNA. Whereas heterochromatin actually is totally opposite to the euchromatin, and it is dark stained region and it is tightly packed and genetically it is inactive. So that is how you can identify. So when you are staining a chromosome, this chromosome based on the stain, what do you say, the stain, how much amount of stain they will take, you can classify the chromatin into two different types. One is called as a euchromatin and the other one you call it as heterochromatin. Okay. So euchromatin, always remember, takes less stain, but it is more active. Whereas heterochromatin takes more stain, it is tightly packed, but it is inactive. So heterochromatin could be of two types. Again, this is of two types. One is called constitutive chromatin, heterochromatin, and the other one is called as facultative heterochromatin. Okay, so constitutive heterochromatin is actually they are permanently conserved or they are condensed. They are condensed. That is not changed from heterochromatin of the DNA sequences. Okay, then thick and condensed state of constitutive heterochromatin. Chromatin, it replicates late in the S phase. So what we can say is now when you are staining a chromosome, that is a chromatin, what it will do, it will take a part of the chromatin will take less stain and a part of the chromatin will take less stain. So the one which takes a less stain, you call it as a euchromatin, which is very active, and a part of the chromatin or the chromosome which takes up more stain, but more stain is called heterochromatin and it's generally inactive. So this is about heterochromatin and euchromatin. Now we can, again, they said heterochromatin is divided into two types. Just now I told you one is constitutive and the other one is called as the facultative. Now facultative heterochromatids are unstable, easily changes to euchromatin. Whereas heterochromatic regions could be easily recognized on chromosome structure in the form of chromomeres, chromocenters, and the knobs. Now chromomeres are what? Chromomeres are nothing but actually these are all staining mechanism and everything they have done, that is a preparation of the chromosome plate to study the chromosome structure in detail. Unless until you stain them and unless until you prepare a plate, you cannot actually go and know the exact what is it structure as well as the shape of the chromosome. So the staining is very, very important. For that reason, we need to stain them and try to identify what are the different parts of the regions that take up the stain and then don't take up the stain. So what they're saying is facultative heterochromatins are unstable. Okay, and these heterochromatic regions actually they are easily recognized because they take up the dark stain. No? So they can be easily recognized on the chromosome structure and in the form of what? Chromomeres, chromocenters as well as the knobs. What are chromomeres? The chromomeres are nothing but they are all regular features of all prophase chromosomes, but their number, size, distribution, and arrangement, they are specific for a particular species. Now, they are the regions actually with varying size near the centromere. Next is, then what are knobs? Chromocenters actually are the regions with varying size, whereas knobs are considered to be the spherical bodies or the regions with spherical in shape. So these are nothing but they are the structures which you can identify, okay, in the heterochromatin region. So now we know what is euchromatin and what is heterochromatin. Now we'll go to the actual discussion, that is karyotype. Just now we have told you karyotype is nothing but a summary. Okay, it is a summary of the genetic constitution or the chromosomes in toto. The chromosome in toto, it may be of any cell of a human being or any animal, say any animal which are arranged. Okay, or you can say an image of the chromosome, an image of the chromosome, summary of the chromosome in an orderly manner. So it is defined as nothing but the study of the chromosome morphology of a chromosome complement in the form of size, shape, position of the primary constriction or the centromere. Secondary constriction, satellite, definitely individually. So what we can say, so what does karyotype do? Karyotype actually highlights the closely or distantly related species based on the similarity or dissimilarity of the karyotypes. Now, you have two different types of karyotypes. One is called asymmetric karyotype and the other one is called as a symmetric karyotype. So this karyotype, what it will do, it not only tells us the size, shape, as well as a position. Okay, it will give you a total summary of, okay, or the image of an chromosome, image of a chromosome of any cell. It might be a human being, it might be of any organism, that is any animal species. Okay, it highlights the closely or distantly related species. So based on this, they have classified into karyotype into two different types. One is called asymmetric karyotype and the other one is called a symmetric karyotype. Now, asymmetric karyotype actually it shows huge differences between the largest and the smallest chromosome. Okay, then whereas symmetric karyotype actually shows very small difference between the largest as well as the smallest. So this is how you can identify. So based on the karyotype, you can also, it will highlight, you can say, the whether the different species are closely related or where they are distantly related. So that is how. So this is about the karyotype. The next is banding technique. Now, what actually you mean by banding technique? No, banding technique actually is nothing but a technique which is involved or which is done basically for identification of the chromosome and its structural abnormalities, okay, in the chromosome complement. So to know, we all know actually the chromosomes, they undergo mutations. Okay, and due to these mutations, what happens? Some chromosomes actually will show the abnormalities. That abnormality may be in the size, it may be in the shape, it may be in the number. Okay, so to identify the chromosome abnormality in a chromosome complement and its structure in the chromosome complement means overall total, then what you need to do, you have to actually go for the banding technique. Okay, it is nothing but a staining technique. So you have got different types of banding techniques based on the type of stain that you are using. They have classified these banding techniques into different types. Now we'll come to that later. So what does it do? It will try to identify the chromosome structure. So the chromosome identification, what it will do, it will depend on their morphological characteristics like the relative length, arm ratio, presence or absence of the secondary constrictions on the chromosome arms. So it is actually a useful tool for the identification of the individual chromosome within the chromosome complement. Now, it is also used for identification of the chromosome segments, okay, that consists of either GC or AT. You all know actually rich regions or the constitutive heterochromatin. Okay, so you can also identify whether these chromosomes actually are of GC or AT. GC means you all know actually the chromosomes, that is the DNA, they consist of the purine, cellulose, pyridines, that is nitrogenous bases. So guanine, cytosine, adenine, thymine. So you can also identify with this technique, that is a banding technique, the chromosome segments that consist of either GC or the AT rich regions. Then next is what on banding chromosome, darkly stained or brightly fluorescent transverse bands. That is positive bands alternate with lightly stained or less fluorescent, that is a negative bands. So on the banded chromosome means whenever you are using a stain to actually go for the banding technique, then what happens is it will take up the stain, no doubt. So some of the region means a part of the region will be darkly stained and a part of the region will be lightly stained. I told you initially about heterochromatin and euchromatin when you are staining. So euchromatin actually will take up the less stain and heterochromatin will take up the darker stain. Here also, when you're actually going for a banding technique, what happens is the darkly stained or the brightly fluorescent transverse bands, okay, alternate with the lightly stained or the less fluorescent negative bands. Okay, so that is how you can know. Now the bands actually are consistent, they are reproducible, and they are specific for each species. So when each species means, suppose if you are going for fish species, say for example, so each species actually show a specific banding pattern. A specific banding pattern. Whether you take an animal, any animal, if you take a fish, or if you take a pig, or if you take any mice or anything like that, or even if you take the other organisms, it will show the specificity. So we can say that the bands are actually specific to each species. So this is called as nothing but a banding technique. So what is banding technique? Banding technique is nothing but it is one of the staining technique which is used for identification of the chromosome and its structural abnormalities. Okay. Now, banding techniques actually also reveal what? It reveals the extensive genetic polymorphism. So it will also show you, it will also reveal the polymorphism. Polymorphism means what? Existing in more than one form, then you say it is polymorphism. So polymorphism manifested as inter-individual differences in the size and the stainability. Okay. So what they have done, initially 4 basic types of banding techniques actually were recognized for the identification of the human chromosome complement. So when they try to actually go for the banding technique, initially they have described 4 different types of banding techniques. What are they? One is called as the Q, C, G, and R bands, and later on 2 additional type of bands also were developed, that is N and T bands, for complete identification of the chromosome complement. So basically, initially only 4 were there, that is Q banding, C banding, G banding, and R banding. I'll tell you what is Q, what Q stands for, what C stands for, G and R, and N and T. So this is, so they were actually based on the type of stain we use, they have been classified. They are named so. Okay. So initially only 4 bands. Later on, they have added 2 more, that is N and T bands. Now these bands actually are widely used in animals also, and all, as well as in the plants, for the identification of the chromosome complement, chromosome aberrations, that is abnormalities, and also to find out any the traces of phylogeny. So this is actually called as nothing but, so the banding techniques are nothing but the staining techniques, and they have been what is it introduced only to know what the abnormalities, to study the structure, to study the size of the chromosome complement, and also to study the various chromosomal abnormalities. Okay. Now, classification or banding of the chromosomes. So, banding techniques actually fall into the following 2 groups, major 2 groups. One is called bands distributed along the length of the whole chromosome. So, when the bands are distributed along the whole length of the chromosome, in that they have given 3 types. What are those? One is GIMSA banding. Just now I told you, G banding. So, G stands for GIMSA. So, it is called GIMSA banding because here the stain we are using is GIMSA. So, it is called as GIMSA banding. Then the Q banding, Q stands for what? Quinacrine. So this is again a stain. Quinacrine is a stain. So that is why it is called as a Q banding. Then the third one is called R, that is a reverse banding. So these are, so in this in this category, actually 3 types of banding will come. So that means when you are using these 3 different types of banding technique, the bands are distributed along the whole length of the chromosome. Then the second category is bands that stain specific chromosome structures, only specific chromosome structures. Then they are divided into 4 types. What are these? One is centromeric heterochromatin staining. Just now I have told you, what is C banding. C banding is nothing but centromeric heterochromatin staining. Means it will take up, it will stain only the region. Centromere is present in the center, no, and in that also the heterochromatin region. So that is why it is called C banding. The next you have N banding. I told you, so that is also called as NOR staining. That is nucleolar organizer region staining. Nucleolar organizer region staining. Then you actually, this is present, this is a part which is present in the centromere or the kinetochore. Then you have got a T banding, okay, and then the last one is the FISH, that is fluorescent in situ hybridization. It is called fluorescent in situ hybridization. So the classification of banding of the chromosomes. So based on this, they have, I mean, bands distributed along the whole length. They have under that 3 banding techniques have been included, and the bands that stain only the specific chromosome structures in these 4 techniques actually have been included. Okay. Now, next to come in. So this is the banding technique, different types, characteristics of various basic banding techniques. Okay. The next, so here see Q banding. This is Q banding. Here you are seeing the band. Okay. Then you have got the C banding. Okay. Then this is called as the G banding. Then here you have R banding. Then this is the T banding. Then you have N banding. Now, this is how the chromosome model they have given. This is a chromosome structure. Okay, so initially, the chromosome structure will be that, like this. It is just a fibrillar network like. Now, banding pattern of Q, G, R, and C bands on the human chromosome complement. So, chromosome B and C, chromosome band C and G clearly identifies the secondary constituents of the chromosome number, whereas C band stains and identifies pericentromeric region. So each banding actually specifies a certain region. Okay, specifies a certain region on the chromosomes. Okay, so like that, C and G identifies the secondary constriction. So they say that C band clearly stains and identifies pericentromeric region. Okay, then both C and Q bands actually are equally important for staining the distal part of the long arm of Y chromosome. Then you have Q band. Now, partial Q banding was reported for chromosome 3, 13, and 21. Okay, so 3-letter coding system actually for banding procedure has been given. So we can say that actually these different banding patterns, okay, of Q, G, G, R, and C on the human chromosome, which will actually clearly, what does it do? It will identify the centromeric region. Okay, not only the centromeric region, but also the other parts of the region. One is a distal. So each band actually has got a specificity, and it will stain only a specific part or the specific region of the chromosome. Now, see, Q banding, that is quincarin banding. Here, the band stains the chromosome with fluorochrome, that is quincarin mustard or quincarin dichloride, okay, observed under fluorescence microscope. This is about the quincarin. So, they have given how we have to do this. So, 80 rich regions. The next, you have C banding, that is called constitutive heterochromatin banding. C banding is called constitutive heterochromatin banding. And here the centromeric regions with constitutive heterochromatin, where satellite DNA was located, actually are stained with this particular type of banding. Okay. And the C banding technique is based on the denaturation and renaturation of DNA. And the C bands are polymorphic in size. So it also helps in analyzing the abnormalities in the centromeric region. So this is about the C banding. Then you have GIMSA banding, which is called as the G banding. So it uses the proteolytic enzyme trypsin. So G banding actually uses the proteolytic enzyme called trypsin as a pre-treatment, followed by staining with GIMSA. So it when you are after banding technique, all these you have to visualize under the different types of microscopes. So for GIMSA banding, you have to visualize that banding pattern under a bright field microscope. So here GIMSA positive bands, that is the dark bands, are AT rich. Okay. Whereas the GIMSA negative bands, light bands, they are CG rich. So we can detect the abnormalities by G banding. So like this, I mean, when you are using these different types of banding techniques, the main what is the logic behind using these techniques is nothing but to not only to find out the banding technique in different regions, but at the same time to find out the structure, to find out the abnormalities actually that occur. So by banding technique, by using different banding techniques, you can easily identify the various chromosomal abnormalities. So that is why they have adopted these different banding techniques. The next you have R, that is reverse banding technique. So reverse banding technique, also called as the R banding technique, it is AT rich region patterns, and these are all based on the thermal treatment of the chromosomes. And reverse of Q, this is actually basically it is a reverse of Q and G banding technique. And here this will allow only the observation of minor abnormalities in the chromosome structure, that too in the terminal regions of the chromosomes. And this technique actually is performed on a fixed chromosomal preparation and is based on heat denaturation of chromosomal DNA. And it is observed, this R banding technique, okay, that is GIMSA stained R banding technique, can be observed only under phase contrast microscope. It occurs only under phase contrast microscope. Now, whereas actually see different types of microscopes you have, like you have the compound microscope, basic compound microscope, we call it as light microscope. Like that, other light microscope, you have different categories, like confocal microscope, polarizing microscope, then you have phase contrast microscope. Okay, so different types of microscopes you have. So they are saying that the GIMSA stained band should be observed only under the phase contrast microscope. The next T, that is telomeric banding. Now T bands are in fact the segments of R band that are most resistant to the heat treatment. So here the clear marking telomeric region. So it will stain only the telomeric. T means it stands for telomeric. So where is telomere located? It is actually located at the towards the tip of the chromosome. So here the clear marking of the telomeric regions of the chromosome with T banding enables us a detailed analysis of the structural arrangements, rearrangements at the ends of the chromosomes. So it allows the detection of the human chromosome 22. So this is about the telomeric banding. Then you have N, that is nuclear organizing region banding. So nuclear organizing region staining, that is NOR staining, is a technique that stains the NOR regions, okay, that contains the genes for ribosomal RNA. Actually, NOR are located in a satellite stalks of acrocentric chromosome. And these acrocentric chromosomes actually have log. So by using this technique, only the NOR regions are stained by using this technique. And here you use a specific staining technique which is called as silver staining, Then, so this is the chromosome banding techniques and their application. Just now we have gone through. So different types of banding techniques. See, Q banding, G banding, R banding. Then you have replication banding. Then you have C banding, NOR banding, like that, different types. And what is the type of stain that you have used, and what is the microscope they have used also they have given, and what are the uses and advantages of using this particular banding technique. So almost each and every banding technique has got specific use and specific advantage. So this will help you, or this will give you not only the structure, detailed structure of the chromosome, but at the same time, it also help you to detect the various abnormalities that occur in the chromosome. Now, another one is sequential banding. Now, sequential banding actually is the modern banding technique that they have given. So here the routine cytogenetic analysis or diagnosis, they say a single banding technique is usually sufficient for detection of the chromosomal abnormalities. But sometimes what happens is more complicated chromosomal rearrangement occurs. So at that time, what happens is often require sequential staining. So when complicated chromosomal rearrangement occurs, then you need to actually go for a sequential banding. Now, if you this sequential banding actually will what is it? It will tell you, okay, give you a clear-cut picture. So here, why they use this? Here, what they are saying here is the sequential staining of these say metaphase chromosome. You all know actually when you are going for the staining mechanism, any type of staining if you use, the chromosomes actually undergo different types of phases. That is prophase, metaphase, anaphase, and telophase. So basically speaking, actually in the metaphasic stage only, the complete chromosome structure can be easily identified, and at this particular stage only, the chromosome will attain a size as well as the shape. So it has a fixed size as well as a fixed shape. Whereas if you take a prophase stage, here the chromosomes actually will be preparing itself for the division, but it will not be actually attaining a particular size or a particular shape. So that is the reason why when you are actually preparing a chromosome plate or to study the chromosome structure, you need to actually inject colchicine, which will arrest the metaphasic stage. So in the metaphasic stage, why they prefer is because in the metaphasic stage, the chromosomes actually attain a fixed the shape as well as a fixed size. So that where there will be a very clear cut the picture of the chromosome structure. So that is why only during the metaphase you can easily study in detail about the structure of the chromosome. So what they are saying is, so sequential banding when you use is sometimes a single banding technique actually will help you to detect the chromosomal abnormalities. But when there occurs more complicated chromosomal rearrangements are there, then that time what you need to do is you have to actually go for a sequential banding. Now, the sequential banding technique, sequential staining of the similar, but several banding techniques, and the process is known as sequential banding. Means 2 to 3 times you have to repeat that banding technique. So that is called sequential banding. And here, in the quality of the chromosomes in this banding, sequential banding will deteriorate with each staining. Therefore, it restricts the sequential banding up to 3 or 4 different staining techniques only. So what you mean is you cannot go beyond. So at least 2 to 3 staining techniques you can go for, but not beyond that. If you go for banding technique, or if you use 3 to 4 technique or more than 3 to 4 techniques simultaneously for that particular chromosome, then it will totally deteriorate. It will deteriorate the complete banding technique, and you will not be able to identify not only the structure, but at the same time, you cannot be able to identify or study any abnormalities. So that is the reason why sequential banding, no doubt, okay, you have to go for it, but only in the cases when there are complicated chromosomal rearrangements. But there is a limit for it. You can restrict the sequential banding only up to 3 to 4, but not beyond that. For example, single metaphase. Okay, then first procedure you are following, then you go for Q banding. Then second procedure you go for G banding. Third procedure you go for C banding. Then later on what happens, it deteriorates the chromosome quality, and therefore it restricts up to 3 or 4 staining procedure. So that is why you should not go beyond that limit. Okay. The next is simultaneous banding. Now, simultaneous banding actually is a technique that produces simultaneously 2 types of banding on the same metaphase or on one slide, but different metaphases. Okay, so this is actually, so simultaneous banding you can go. So this is a technique that you can use it, okay, simultaneously 2 types of banding on the same metaphase or on one side, but different metaphases. So for example, same metaphase. Then next you have the first procedure, G banding, second procedure, C banding, or a single slide with different metaphases. So means you can actually, when you are actually seeing the slide under the microscope, you have to select 2 different metaphasic states, not one single metaphase. So like that you can go. So that is how you have to go for simultaneous banning. And here also simultaneous banning, it will restrict up to only 2 staining procedures. Beyond that, you should not do. Again, it will try to deteriorate the chromosome structure. Okay, differential staining along with longitudinal axis of the mitotic chromosomes is referred to as chromosome padding. Okay, so chromosome padding techniques, what it will do, it will try to produce a series of consistent landmarks along the length of the metaphase chromosome that allow for both the recognition of the individual chromosomes within a genome and also identification of the specific segments. And the first chromosome banding technique, they're saying that was first given by, okay, Mary Law Pardue and Joe Gall. Okay, so here actually they have made the first chromosome banding technique, or they have introduced the first chromosome banding technique, and that too the chromosome preparations from mice. So they have taken a mice, and when they prepared the chromosome preparation of the mice, they have first introduced this chromosome banding technique. Later on, it was gone. Okay, so only here, only the centromeric regions, when they introduced this technique, what they did is, when they tried this technique, it could what is a stain only the centromeric region. So that is why it was called as a C band. It is called as a C banding technique. Okay, then you have G banding technique. Okay, so in G banding, the chromosomes are subjected to controlled digestion. Q banding technique, already we have seen this. Okay, then R banding technique, T banding technique, C banding technique. So different types of banding techniques that we are using. Next, again, you have characteristics of euchromatic bands in mammalian chromosomes. Now, in mammalian chromosomes, if you see G bands and as well as the R bands. Okay, so this is only a basic this thing structure. See here, the procedure for making a human karyotype. So how they have done, okay, how they have prepared. See, this is a chromosome plate that they have prepared and they are observing. Okay, so the slide is viewed by a light microscope. With the light microscope, when you are visualizing, you observe like this. Then what you have to do is, then you have to take a, I told you picture. After taking a picture, when you are observing this picture over here, are they arranged? They are not arranged in a sequential manner. So what you have to do is, you have to take a picture, cut the, what do you say, the different chromosomes according to their size, then take another paper and then you have to paste it. Okay, so karyotype actually will give you only an image. It will give a summary, whereas karyogram will give you what? It will give you a clear cut, what do you say, idea based on the size and all those things. Okay, this is the procedure you have to follow. So the slide is good. See how they have done here. A sample of blood actually is collected and it is treated with the chemicals that stimulate the cells to divide. I told you, colchicine has to be added because what happens? It will disrupt the spindle formation. It will disturb the cells in mitosis, where the chromosomes are highly compacted. So the cells are then subjected to centrifugation. So what you have to do is, you have to take a sample, any sample. You may take a blood, you may take you may take a blood sample, or you may take any other tissue also, you can take it, and then you have to treat it with the chemicals, and then you have to add the colchicine to it. The colchicine will disrupt or the cell division, the spindle formation. It will stop the cells at the mitosis stage only. Then you have to centrifuge that what do you say, the collection, whatever you have taken. After collecting it, what you have to centrifugation, what you have to do is, you'll get two things. One the pellet, and the other one is called as a supernatant. Now when you take this pellet, will be at the bottom. Pellet is also called as residue, and the upper liquid form you call it as a supernatant. Then you have to discard that supernatant, and you have to take only the cell pellet, that is a residue, and then you have to suspend it or put it in a hypotonic solution, and this will causes the cells to swell. Then what you have to do is, next the sample is then again centrifuge. You have to go for centrifugation second time to concentrate the cells. Then again the cells are actually suspended in a fixative. They are stained and then they are placed on a slide. After placing it on a slide, then what you have to do, you have to observe it under the microscope. So this is a preparing the cells for a karyotype. So when once you prepare the cells, you have to visualize it under the microscope. After visualizing it under the microscope, you all know that they are not properly arranged. Take the photograph. After taking the photograph, cut the individual chromosomes, take another paper and paste it on it in an orderly manner according to their size and according to their shape. So that is how you have to do it. So this is the procedure for making a human karyotype. Okay. Then this is the cell structure protocol for standard karyotype. Just now I have told you, the same. See, blood cells collected in sterile tubes. then the whole blood leukocytes actually are separated from the red cells. They are to do with culture media. Then mitogen. Okay, you might also use a colchicine. Here mitogen you have to add because to arrange the metaphasic state, that is PHA, phytoheme agglutinin has to be added. Okay, then cultures you have to incubate the culture and then shake, and then you have to add the colchicine to the cultures. Okay, and then you have to do it. This is the procedure that they have given. Then cell culture protocols for standard karyotype. Then you have to tube centrifuged after incubation for 72 hours. Supernatant discarded. You have to take the cells, resuspend the cells in hypotonic treatment. Then again, centrifuge the tubes, discard the supernatant, take the pellet. Then you have to fix that cells in a carbonized solution. Then cell suspension should be dropped on a clean slide to fix the chromosome. Then keep the slide at a room temperature, okay, or in the oven, and prepared slides have to be steamed or banned. Go for the banning technique. So same way again, similarly, different types of banning techniques I told you know. For quinacrine banning, you have to use this. So materials required here: air dry slide of metaphase chromosomes, twinkle staining solution. All these you need to have to go for Q banning. If you want to go for, this is how you have to do. Place air dry. This is the procedure, and those are the materials required. Then next is GTG technique for G banning. So that was for Q banning, and this is for G banning. So material required is HBSS, trypsin solution, 70% and 90% ethanol, 2% GIMSA, and H slides of metaphase. That is xylene. Then GTG staining for G banning. How you have to do. Okay. Then for G banning, then next is the same. Then centromeric heterochromatic stain, that is C banning technique. What are the materials required? Then next, what are the procedure? How you have to follow. This is all the procedure. The next, reagents and solutions for C banning. The nuclear organite is a reagent staining, that is not staining. What are the materials required? The next, how to go about with the procedure. Then reagents and solutions that are required for not staining, that is GIMSA solution, silver nitrate, the gelatin solution, silver nitrate solution. The next is the reverse banding. In the reverse banding, R banding again. What are the materials required? Then sodium phosphate buffer, air-dried slides, then GIMSA staining solution, cupping jars, and circulating water bath. These are the things required. Then this is the procedure you have to follow. So this is all about the different types of banding techniques. Now today we know, so what is the chromosome, how to study the chromosome, because to study the chromosome, to know about different types of chromosomes, it is very, very important for us to prepare what? To prepare a chromosome plate. So how to prepare a chromosome plate, just I have told you. So you may take any sample, okay, you may from the human sample or any animal sample, you can take it, and that to sample means blood sample you may take or any other tissue you may take it, and you have to follow the procedure protocol. After following the protocol, you can stain them by using any of the banding techniques. And once you prepare that slide, what you have to do is you have to take the photograph, observe it under the microscope. I told you, know, after observing the under the microscope, you all know actually the chromosomes will not be arranged in an order. So what you need to do is, when once you observe it under the microscope, take a photograph of it. After taking the photograph, take another paper and then cut those chromosomes in pairs and arrange them in an orderly manner according to their size and according to their shape. So when you do it, what happens is easily you can identify how many chromosomes are there, you can identify, and at the same time, different sizes of the chromosomes also you can identify. So that is how you have to actually go about with the chromosomes. So different banding techniques actually have been given. You can follow any of the banding technique, and each banding technique actually will stain a specific region. It will stain a specific region. So when you are going for a specific banding technique, easily you can know that particular what they say region is good, or it is actually having any abnormality or not. So easily you can identify the various chromosomal abnormalities that are present along the whole length of the chromosome or at any part of the chromosome. So this is how we have to go for the what do you say, the chromosome preparation of the chromosome plate to study the chromosome. That is, so this is all required for the study of the chromosome. That is what we call it as nothing but a karyotype or a karyogram. Next is we will talk about the chromosomal inheritance. Now, what do you mean by a chromosome inheritance? So, chromosomal theory of inheritance. One second, I will try to share the. Okay. Now, this is about the chromosomal theory of inheritance. So what do you mean by inheritance? Inheritance is what? Nothing but the transmission of the characters. Okay, from one generation to the another generation, or you can say transmission of the characters from the parent to the offspring. So that is what we call it as nothing but. So what do you mean by chromosome theory of inheritance? And what do you mean by sex determination and the inheritance of the sex traits? How actually the characters from the parents are going to their offsprings. This is what we have to see. So this has been divided into three different categories. One we call it as chromosome theory of inheritance. Next is chromosome mapping, and another one is sex determination and the inheritance of the sex-linked traits. So actually, the credit goes to Gregor Mendel. You all know actually Gregor Mendel actually is the father of genetics. So he has discovered the laws of heredity and states that he states that or he came to the conclusion stating that the hereditary principles without the knowledge of genes and chromosomes. So actually, what he was said now he has actually given the cytological basis of the hereditary. So the chromosomal theory, basically, or the chromosomal theory of inheritance, was based on the laws of heredity, which were actually given by Gregor John Mendel. So Gregor John Mendel, actually, we call him as nothing but a father of genetics. He actually, you all know, worked on pea plant. First, he worked on the pea plant. Later on, actually, he has worked on the animals. But initially, he worked on the plant and he gave different laws of heredity, like you have law of dominance, law of segregation, law of independent assortment. So in these different laws of heredity, he told what is a dominant character, what is a recessive character, and what do you mean by F1 generation, what do you mean by F2 generation. And when you are crossing the generation, one generation to another generation, how actually the different characters are inherited from the parent to the offspring or from one generation to the another generation. So the credit goes to him. That is the reason why he is actually called as the father of genetics. And based on his laws of heredity only, the other scientists actually have tried to work. Okay, and then they have worked on the different aspects. So later on, after Gregor John Mendel, now you all know actually Friedrich Mischer was the first one researcher actually who to isolate and identify the nucleic acids. So he has given the nucleic acid. So what are the different types of nucleic acid? That is nothing but your RNA and DNA. So here now, Friedrich Mischer was the first person, or he was the first researcher to isolate and also to identify the nucleic acid. So later on, in 1878, Walter Fleming was the scientist who described the chromatin threads. That he said that there are chromatin threads which are present in a cell nucleus, and also he tried to actually give us the information regarding the behavior of chromosomes during mitosis, how the chromosomes behave during the mitotic cell division. Later on, August Weismann was there who described the role of meiosis for reproduction and the heredity. So that is how we have given. And in 1888, Walder actually coined the term chromosome to coin the term chromosome. So likewise, different authors actually have put their views regarding, so not only after we can say the Gregor John Mendel, various other scientists have come forward and they try to do the research and they have actually given their findings. So these were the different scientists actually who have given their findings. Now, you all know here, coming to the mitosis and meiosis. So you all know basically there are two different types of divisions, that is mitosis and the meiosis. Mitosis actually is a somatic division which occurs actually in the somatic cells, whereas meiosis is a reduction division which occurs in the germ cells. Okay. So, and during this particular division only, before the cell divides, Now the chromosomes actually have to prepare the nucleus. You all know actually will prepare itself for the division. So before the cell division, actually that is either mitosis or meiosis, the chromosomes will not be arranged. So they need to be arranged. So they will actually they are in the form of fine threadwork like structures. It is formed in the form of a chromatin network where you will not be able to identify the chromatin network. So only at the time of division only the chromosomes will attain the shape. So this is one thing. Then later on, certain Theodore Boveri and Walter Sutter, these were the two scientists actually who tried to give the chromosomal theory of inheritance. So their observations actually, that is Boveri and Sutton, led to the chromosomal theory of inheritance. So today we say that actually the chromosomal theory of inheritance actually was identified by Sutton and Boveri, who they said actually the chromosomes are the genetic material which are responsible for the Mendelian inheritance. So now what does the chromosomal theory of inheritance suggest is they say that during meiosis, the homologous chromosomes they migrate as discrete structures that are independent of other chromosome pairs. So certain laws they have given, and the second one what they said is the sorting of the chromosome from each homologous pair into pre-gametes appear to be random, and each parent will synthesize the gametes that contain only half of the chromosomal complement. Even though male and female gametes, that is sperm and the ova, egg differ in size and morphology, they have the same number of the chromosome. So these are the different, what do you say, the aspects that are given by Sutton and Boveri under the chromosomal theory of inheritance. Okay. Now the gametic chromosomes, what do? They combine during fertilization to produce the offsprings, and these offsprings again with the same number of the chromosome. So you all know actually they say we have the same number of chromosome, humans, from the parent to the offspring, or in different what do you say, humans, if you say the number of the chromosome is the same, because we all belong to the same species. So that is the reason why we all have the same number of the chromosomes. Now later on in 1909, Johansson actually introduced the term gene. Now he has introduced the term gene. So what is a gene? Gene actually is nothing but it is a part of the chromosome, or it is present in the chromosome at a specific point. So chromosomal inheritance was proposed long back, okay, before the what is it? The genes were introduced. Now this chromosomal theory of inheritance actually was supported by many of the evidences. So that is why today when you're talking about the chromosomal theory of inheritance, most of the evidences actually are very true, and they have actually given the correct information regarding the genes. So they studied and they tried to explain the following questions. Like they said that actually, according to the chromosomal theory of inheritance, that is Sutton and Boveri, they have given importance of the chromosomes, and they said that actually the chromosomes work as nothing but carriers of hereditary information. And they say that actually in this chromosome the genes are present, and these genes undergo linkage, and also they undergo crossing over, and also the chromosome mapping, and also they have described about the sex linked inheritance. So later on, actually in the year 1900, William Bateson and R.C. Punnett, they were studying the inheritance of the sweet pea, that is a pea plant, and they studied two genes. One was affecting the flower color, and the other was affecting the shape of the pollen grains. So when they tried to actually cross these pure lines and so they could obtain heterozygotes in the F2 generation. So like that, and this actually they have shown in the form of a square. That square we call it as nothing but the Punnett square. 1 second. So like this, they have two phenotypes. So this is how. So these are the different, what do you say, the scientists who have given. So the confirmation of the Bateson and Punnett's hypothesis, actually, they had to await the development of Drosophila as a genetic tool. So later on, after the pea plant, that means the pea plant actually was studied by most of the scientists after the Gregor John Mendel, and they have came to some conclusion. Then the next model that they have used actually was nothing but the Drosophila. So Drosophila actually was extensively studied as a genetic tool, and the person actually who studied or who did a lot of research on these Drosophila was Morgan. So Morgan actually started his work with Drosophila melanogaster. Now Drosophila is nothing but a fruit fly. He chose this fruit fly because he said that they can be cultured very easily, and they are present in large number, and they have a short generation time. So they can be breed. They breed very rapidly, and you can have them and more accessibility is there, where you can get it very easily. And they have only 4 pair of the chromosomes, and the polytene chromosome can be identified under microscope. So that is the reason, and they said that it has clearly marked characters. So that is the reason why they have chosen this particular animal as their model, and they try to exploit this animal by doing lot of research. Okay, so first was the pea plant, and later was the Drosophila melanogaster. So they have taken different, what do you say, the characters, like the color of the eye, shape of body color, shape, and all those things, and they try to cross this, and they try to actually give us not only gave us the information regarding the F1 and F2 generations, but they also told us when we are crossing the going for the test cross. Okay, what are the characters that are inherited? So they also studied the importance of the test cross. The importance of the test cross, they use the test cross because when one of the parent actually is crossed against one of the recessive allele, that is the offspring, what is the result, net result you get it. So here they have actually mentioned the different types of crosses, not only the cross between the what do you say, the different parent, but their offsprings, parent and the offspring, and also they try to cross between the parent with one of the offsprings, and they have given us the various what do you say, the suggestions. Now how they actually couple with each other, and coupling will take place, and how the repulsion phenomena will take place. Then they have also talked about the crossing over. What do you mean by crossing over? Crossing over actually is nothing but when the homologous chromosomes come in pairs, that is in meiosis, what they do is they try to exchange their parts in a process. That process we call it as nothing but the crossing over. This crossing over, they say that occurs at a specific point called as the nothing but chiasma. So chiasma actually is a point, okay, where actually the crossing over takes place. Now, what do you mean by crossing over? Crossing over is nothing but the exchange of material between two homologous chromosomes. They have given different aspects of the things. Then after crossing over and the chiasmata phenomena, then they said that actually when two genes are close together on the chromosomal pair, they do not assort independently. So that is what. Now we'll try to give a brief summary of the chromosomal theory of heredity, or the chromosomal theory of inheritance is. What does actually the chromosomal theory of inheritance or the chromosomal theory of heredity talk about? They say that the genes are located on the chromosomes in a linear order. Okay, that is, they are arranged in a linear order, and each gene occupies a certain place on the chromosome. That particular place you call it as a locus, or you can say each gene is placed, or each gene actually situated at a specific point on the chromosome, which is called as locus. And each homologous means what? Same. Each homologous chromosomes contains only one allelic gene from the allelic pair. And genes of one chromosome, they form a group of linkage and are inherited together. Okay, and the number of linkage groups will be equal to the number of the haploid set of chromosomes. And the linkage of genes can be interrupted during the meiosis, either by crossing over phenomena, that is recombination. Okay. Then the percentage of crossing over will be directly proportional to the distance between the genes. And the distance between the genes on the chromosome can be estimated from the proportion of the recombinant offspring. So that is what they have given us the different aspects. Okay. The next is what Morgan also. So for all these his discoveries, you all know actually Morgan, who worked on the Drosophila melanogaster, he was awarded the Nobel Prize, the Mendel. So he was also awarded the Nobel Prize for his discoveries concerning the role played by the chromosomes in heredity. Then genetic linkage also occurs. Then examples of complete linkage and incomplete linkage they have given. So they studied in detail about the linkage phenomena as well as the crossing over phenomena in the Drosophila. So this is all. Then significance of crossing over. Okay. Then what do you mean by genetic map or the linkage map? So a study of linkages between many genes enables the creation of a linkage map or a genetic map. So what does genetic map or linkage map see? So you have actually in a gene, linkages will take place. The study of linkage between many genes, because on a chromosome you don't have one single gene, you have a number of genes. So all put together, study of the linkages between many genes, it will constitute nothing but a linkage map or a genetic map. So genetic map is nothing but an arrangement of genes on the chromosome. So this is how they are given. And they have postulated that genes are arranged in a linear series, same way. The genes which are close will be separated by crossing over. Then also you can find out the frequency of crossing over. Okay, then and the distances between any two genes you can measure by using